Good afternoon. I am here to talk today about what seems like a really modest topic, rapid health protection for the population. And it seems like that would be something that is done or that we are already doing today with the field of vaccinology. And I'm trying to create the case that we are not effective at that we need a new capability, that the capability must be disruptive, and the method is going to use tools from biotechnology and gene therapy to be able to achieve that for the population. So I am a clinical geneticist. I see patients once a week in the field of cancer genetics, and I see firsthand the benefits of genome technologies and what they have done for the care of patients. I've also been somewhat of a voyeur of gene therapy since my time of training at NIH, watching that community try to create a capability that allows you to put a gene inside an individual that has a broken gene in their whole body or a missing gene in their whole body and have that gene produce that protein at a level that will correct that problem for them. So with gene therapy, the goals are to permanently correct or replace genes to treat a severe or inherited disease. And typically the method that is employed is to use a virus. We either use a virus to cut into our genome and permanently place a gene into our body, or we can put a virus inside the nucleus of our cells, and that virus then can produce the protein. The challenge, again, with gene therapy is that you often need to put this into many or all the cells of your body, and that virus can lead to, if it cuts it in an inappropriate site of the genome, it can lead to inadvertent cancers years later, or the proteins itself that make the virus can lead to immune reactions that we don't want. So the methods that I want to look at are how can we see the effect of gene therapy without using the virus, and how can we use this for healthy people. And for healthy people, I don't want that protein to be made permanently in my life. I want it to be transient. I want to be able to control it. I want it to be present when I need it, and then I want it to be gone in case there are untowards or unexpected effects of that gene. And I want to use it to prevent or treat acquired disease, but particularly to prevent diseases. And there's two methods that I want to show that I think that we can employ to do that, so that we have genes used as gene protections that are transient, that are able to produce proteins, and that are ephemeral. The first I will talk about is the ability to gene encode monoclonal antibodies. And just for a moment, let's admire what I think the problem is. And we've seen a lot of discussions about what problems we're trying to solve today. If we look at vaccines, vaccines often take us decades to develop. For some diseases, such as malaria, we've been working on it for decades, and we do not have a vaccine. There are only several dozen vaccines that are commercialized, used, and licensed in the United States today. And even for a vaccine where we are effective, such as influenza, and we have global capability with multiple major biopharmaceutical companies and biodistribution that is global, the vaccine itself is not achieving the result that we have intended it to. So as an analytic framework, let's look at the H1N1 2009 outbreak that started in Mexico, spread to the United States. The data that you're seeing on the bottom right-hand side of the slide is from the CDC modeling the outbreak, and what you're seeing on the y-axis is millions of cases of influenza-like illness and hospitalizations per week. And then what you're seeing along the x-axis is these number of cases over a period of time. And when we make a vaccine using our latest technology for flu, where we know how to make vaccine, it takes us 6 or more months to produce the vaccines after we've identified the sequence from the index case or from cases that have evolved over time. By the time we ship that vaccine and put it into a human body, you can see many of the cases have already been transmitted. It also takes an additional amount of time for that vaccine to achieve its effect. The vaccine begins an evolutionary process within one's body that is a semi-stochastic process of cellular signaling, handing off signals that will ultimately lead to what we are trying to achieve, which is protection with a biomolecule called an antibody. And for influenza, we know that it takes at least 2 weeks. So by the time individuals have received the shot in their arm from CVS or their local health clinic and had been protected from that influenza illness, in our best case, most of the cases are already transmitted. And this modeling paper from the Center for Disease Control and Prevention showed that we prevented 1.6% of clinical cases in H1N1 2009. 61 million cases could have been averted, and we averted 1 million cases. And this is when we know what we're doing with flu vaccine. More to the point, flu vaccine is only typically 30 to 70% effective each year. So every year, because flu virus changes, we have to develop a new vaccine, which means monitoring it from Southeast Asia, sequencing it, determine which antigenic sequence we're going to make vaccine off of, and manufacturing it. And then as you're seeing in this schematic, yellow is the period of time, notionally, that it would take an individual to develop that protective response, and then they're protected. But the next year, you have to go through this process again of sequence, choose a strain, manufacture, sell and distribute, put the shot in the arm, wait several weeks for protection, and again, and again, and again. And yet, 30 to 70% of those people don't even achieve protection. And what is the ultimate protection that we want them to achieve? The development of antibodies that are going to be able to prevent flu. Now, there are other cellular responses like T cells and so forth, but for prevention, humoral-mediated immunity, the development of antibodies, can mediate protection. The situation is even worse for something like anthrax. It takes 5 doses of commercialized vaccine to be 90% protected. So if we have a situation with anthrax, as an anthrax outbreak, either naturally or in other circumstances, we have to wait at minimum 6 months for people to be protected. And that's when we've already manufactured the vaccine and have it on board. It almost seems like a fool's errand to be providing the anthrax vaccine in an emergent outbreak, because what would be the point if it takes many, many months to have protection? We need a solution other than a vaccine, and we need it to be immediate. Now, as a geneticist, I think can gene therapy help us solve this? Now, what happens in actual people when they get sick with an illness? Again, it's a schematic to keep us all in the same thought process. Assume we have never seen a disease. So on the left-hand side of the screen, you have a naive, immunologically naive individual who is otherwise healthy. They're then exposed to influenza virion or perhaps even Ebola virion, and they are sick. And they either recover or they die. If they recover, they must have generated an appropriate immune response to be able to have them counteract that illness. And in that cocktail of antibodies that are in their body, there is an antibody or several antibodies that are protective against that illness. Can we identify those antibodies? Can we identify them rapidly enough? And can we transfer them to the population? Can we create a transformational event where you go from healthy directly to an immune recovered state, bypassing an illness? Now, this has been known for some time. It was done in the time of the Iditarod. The Iditarod occurred in Nome, Alaska, because diphtheria was occurring, and there was an outbreak in a small town, and the diphtheria antibody that they had to be able to provide to protect people was expired. So they needed to get to the other end of Alaska, I believe Anchorage, and back, and use the dog sleds, and began the Iditarod Challenge. And what their goal then was to do was to get serum to a healthy individual to protect them from diphtheria or to those from sick to help them recover. The problem with direct serum transfer is that you're transferring either, in those days, horse inoculum, which led to immune reactions, or all of the antibody components, which include some that you don't want or need. And their antibody itself, when it is preformed, only lasts in our body for about a week or two, and that antibody degrades. Our own antibodies are constantly being made and degraded. The whole field of biologics has ensued in the past 30 years, where we are able to develop proteins outside of the body in living systems, and it's an enormously expensive therapeutic process where we develop antibodies in vitro systems, grow those antibodies, isolate them, purify them, and often put them in IV bags. And those antibodies then can be delivered to people. as protection. There's one example of this in the anti-infective space, and that's Synagis, and that was made in 1998, where monoclonal antibody is being given to protect infants from respiratory syncytial virus that are preterm. What we would like to do is get the effect of being able to provide cocktails of antibody to naive individuals and then being able to give them to populations. But as I said before, we have a problem with this type of therapy because it's IV, often requires cold chain, and often requires a long time of production process, the fastest right now being about 9 months. Now, we all went through Ebola in the news last year, and we saw with Ebola coming to our shores that really our main form of treatment of Ebola is what it's been for centuries, which is quarantine. Because we did not have vaccine available in time for Ebola, and we had been developing a vaccine for many years. We also did not have monoclonal antibodies on time, and we had been developing monoclonal antibodies in years. It was a question then of scalability. We were unable to make the MAP cocktail of antibodies and deliver them, and we were unable to produce the vaccine and get it out. And the patients did not get Ebola vaccine or Ebola antibodies to any degree for protection. What we did see was that individuals did make it to our US shores. They came to Emory for some cases, Nebraska for others, and even New York for others, as we saw. And this happens to be Dr. Brantley. And what happened with Dr. Brantley is he recovered at Emory, and his blood was drawn and given to other infected survivors as a form of passive transfer, as we just discussed. But why couldn't we give Dr. Brantley's antibodies to everyone, maybe everyone exposed in the villages of Africa where the disease was outbreaking? What would be the problem with that? Well, we would need a lot of that antibody. We'd need production. We'd need cold chain. We'd need IV bags. That's an insurmountable problem with the present technology. So what we're doing with Dr. Brantley's blood and all the US Ebola survivors is we are collecting their blood and we are sequencing their monoclonal antibodies and being able to identify the genetic sequence that makes that antibody. And our goal is to gene transfer the genetic sequence that makes that antibody into healthy people. This is actual data from the Ebola survivors. We can only show you the data that has been shared to the public community at this point. But what you're looking here is a fluorescent activated cell sorting, where on the left side you have a sort from a healthy person. The black dots represent the cells of the immune compartment that are secreting antibodies against Ebola. This happens to be a plasmablast sort. And what you're seeing on the left is none of the cells are in the white box. And the sandbox of the white box with GP positive means the glycoprotein of Ebola. On the right side, you're seeing a sort on a patient that is one of the recovered patients, and they're making plasmablasts that are binding to the GP form that is put in this sorter. And so we are able to very quickly, within days, be able to, usually about 6 days after they have been exposed to illness, be able to sort plasmablasts that are identified to bind to Ebola. Now, we're able to do a myriad number of other things, most of those results not able to share today, where we're able to look at the evolution of that protection of those antibodies, and then we're able to perform functional assays in line with our discovery process, being able to determine which of those antibodies, like a needle in a haystack, are the ones that led to protection. So the future process that we are working on would go like this. When there is an outbreak, whether it is an existing illness that comes every year, such as flu in Southeast Asia, or whether it is a new one, or even a toxin-mediated threat, we would find the infected survivors. We would draw their blood. We would sequence every monoclonal antibody that they're making in their body. We would perform a functional assay on that sequence to determine which ones are protecting. We would then encode that gene either on DNA or RNA, not using a viral carrier. We would put it in a shot, not an IV bag. We would provide it in a form that doesn't require cold chain. We would put that shot into someone's deltoid of their arm, and then their cells would make those monoclonal antibodies, and we would create that transformation that we saw in the schematic at the beginning, where you're going from uninfected, healthy, to protected. So as I showed before, current monoclonal antibodies, they only last about 2 weeks in the human body, and they require re-administration. And that re-administration of serial dosing also would create a problem for immunoprophylaxis. The bottom is what we would like to achieve, where we were able to have 4 to 6 months of duration of that monoclonal antibody by producing that antibody genetically for many months at a time. So let me just show you preliminary data showing that we can do that. On the top, you're looking at actual monoclonal antibodies. This is the ZMEC cocktail that have been designed for mice, and they're being put into mice. And this is a kinetic study showing that there is 147 days of expression of those 3 monoclonal antibodies and the concentration that is present in their blood. And you can see that it peaks around 16 micrograms per ml and is tailing off somewhere between 4 and 6 micrograms per ml. Synagis, which I mentioned before, the trough dose is 38 micrograms per ml and was improved in 1998. Many monoclonal antibodies right now that are in preclinical development for infectious diseases are potent at single microgram per ml levels. So if we are able to achieve a cocktail of 3 monoclonal antibodies at 12 to 16 micrograms per ml at peak and a trough of 4 micrograms per ml at 140, you can see we're within a drug concentration range for protection. On the bottom graph, you're seeing flu, and you're seeing actually each of those antibodies plotted independently, each one expressing somewhere between 2 to 4 micrograms per ml. This is courtesy of David Ho from Aaron Diamond here in New York City. However, we want to make sure those antibodies work. And so when we look at the antibody production of this in a mouse challenge, this happens to be a flu challenge. This is a mice experiment where the experimental conditions are the mice has been designed to be sensitive to influenza virus. They gave a strain called H3N2. The control on this Kaplan-Meier curve is the gray line, and what you're seeing by 9 days is the plot of survivor, all the mice die. The mice that were given the monoclonal antibody, both at high dose and low dose, that cohort of mice survived, and at high dose, none of the mice passed away. And in fact, none of the mice actually had weight loss. So that is one method that we think we can perform DNA or RNA-based gene-mediated immunoprotection. Another method that we have begun to think about is how to use this with red blood cells. And what is unique about red blood cells is that they do not retain any of their genetic information after they're mature. And so this thought of using this was derived for a program where we could take advantage of the fact that the red cell lasts in the human body for about 120 days. So now I have a carrier that is going to carry my drug of interest for 120 days, but I have no genome. But maybe that is a good thing. So the notion that we would do in this case is that we would take a naive person, we would draw their blood. In the adult compartment of the bloodstream, there are stem cells that are progenitor cells that will differentiate into blood cells that have nuclei. In this case, we can do what I said was anathema at the beginning. We're going to edit that genome. In fact, we will welcome the academic community to edit that genome in any way that they desire. If that means that they're going to cut the genome near a cancer promoter site, it's irrelevant, because we are no longer going to retain that genetic information. This is going to be a transient state while the cell has the nuclei. The cell will be expanded and differentiated while it has the nucleus, and when the nucleus produces the protein, that protein could either make an enzyme inside the cell or express a protein on the surface, like an antibody that we just discussed. And then it will do something that it naturally does if we achieve our goals, and it will enucleate. That's not trivial, as you'll see. But then we will also add radiation. Now, that may seem like a scary step that we would put into it, but most transfusions in the United States already have irradiation as a component of their process because it is killing T cells, which we don't want for graft versus host in an allogenic donor. And then the result would be expanded, differentiated red cells that are modified for our effect, and maybe for immunoprotection. Now, this program did not begin this way. This program began when I came to DARPA with the goal of trying to make blood that was O negative as a universal donor. so that you could be able to take O negative, perhaps COL Duffy negative, antigens that normally people have immune reactions to, and remove those, and then just simply produce what would be, not remove those, select those red cells that do not have those antigens and just expand them in an in vitro supply. The problem with that program was that it cost, in media alone, over $90,000 to make 1 unit of blood. Through the success of that program over time, and you're actually seeing the dates and plots of that program, by the end of that program, we were at somewhere around $2,000 to produce a unit of red blood cells in terms of number. But the biologic process to do that was not simple. It was actually a DARPA engineering process where much of the goals were on the engineering of the capability, and as you can see, that is actually a pretty incredible plot. But we were missing a couple of very key things. One was the acumen of how to understand that terminal differentiation process and understand all of the steps to be able to ensure those red blood cells matured and expanded effectively. And that's when I met Professor Harvey Lodish to help us with that program and help us with that problem. And indeed he did. But as we were ending that program in that final year, we started to have a conversation around, we've got a problem, we have O negative blood, it's going to cost $2,000, but it's only cost $120 to $240 for me to go to a blood donor center downstairs and give my blood, so I don't have a market. So maybe if we created a therapeutic capability out of the blood, we could add value. And this is what has resulted. And so this is actually Harvey's data where we looked at one of the key problems, which would be if we modified a red cell, particularly with a surface protein that is de novo, what would be the immune reaction to that red cell? So in first of its kind studies, I believe this was published in PNAS, we looked at putting modified red cells back into a mouse when the mouse obviously has its circulating native red cells. The green line is the kinetic plot of the mouse red cells, and you see that introduced cells labeled from a mice, they last about 28 days. That's about the normal half-life. And you're seeing the decay of their native red cells with the green line. But what you're seeing in all of the other 4 lines are surface-modified red cells. And even when re-administered twice, the immune compartment did not develop an adverse reaction to those red cells, indicating that we would have a platform for modifying red cells. So will red cells be a platform technology that to be carriers of any number of medicines, enzymes, proteins, therapeutic proteins, and maybe protective antibodies for future instances that are what we need? That is the goal of our current efforts, and that is why we've invited Professor Lodish, who's helping us with that, to speak to you today to carry on this conversation. Thank you.